Good morning. Hello, everyone. Thank you for coming. I have the privilege of introducing myself. I'm Steve Schaffner. I'm a staff scientist here, a computational biologist, and I've been here for a long time. I used to be part of MPG back in the 1,000 genomes and HapMap era and even before that. These days I work more in pathogens, malaria, and viruses, but I'm still interested in human genetics and natural selection in humans, which is why I'm talking to you today about natural selection in humans. In particular, its effect on human genetic variation and what we can learn from this and how we can detect it. So I think you probably all know what natural selection is. I'll just state it for the record, that it's the principle that alleles that make an organism more successful in terms of survival and reproduction are likely to be transmitted more to the next generation and therefore likely to increase in frequency while they're being selected. And we can, for convenience, we can divide the selection into several different kinds. The first is balancing selection, which is selection that maintains multiple alleles in the population at some intermediate frequency. And there's purifying selection, which is the elimination of new mutations that are deleterious. And finally, there's positive selection, which is selection for some beneficial trait. And actually, the last two are really sort of flip sides of the same thing, because if you're selecting for one trait, you're selecting against some other trait. You have to be choosing one. But it's convenient to distinguish them based on what's starting out as rare. You can think of purifying selection as eliminating new rare things. And I'll start by talking about balancing selection. This is probably the rarest kind of selection. It's kind of cool when you can find it, but it's a little difficult to detect and probably doesn't happen very often. There is one very well-known case in humans, the sickle cell trait, sickle cell allele, which was one of the first cases, probably I think it's the first case of identified natural selection in humans. It was, I think Haldane in the 50s noticed that certain diseases involving hemoglobin were much more common in places where there was a lot of malaria. And he hypothesized that there was natural selection was playing some sort of role. And he was correct. And these two maps, the top map shows where malaria occurs worldwide, and the bottom map shows where there's a high prevalence of the sickle cell variant, the sickle cell trait. And the reason is quite clear. You can, it's easy to find out. To check, you can actually measure the difference in fitness. If you have one copy of, if you're heterozygous, if you have one copy of this allele, then you have considerable protection against malaria. So if you have no copies, you're exposed to malaria, are likely to die younger, because malaria is a big killer. If you have one copy, you have a real benefit. If you have two copies, then you get sickle cell disease, and you're also likely to die young, because without modern medical care, it's a very serious disease. And so you can just look at the survival. This was without that kind of medical care. If you look at the survival, it's very clear that heterozygotes in this case have an advantage. And that means that there's selection pressure to maintain this allele at some intermediate level, so that the maximum number of heterozygotes in the population. It's not a very pleasant solution, but it is a sort of evolutionarily stable solution to a severe selective pressure. You can get balancing selection in other ways by selection for diversity. There are a lot of cases in which it's good to be different from other members of your species, like escaping from predators. If the predator is used to finding purple people and eating them, if you're green, it can be kind of good to be green then. And it's quite common in resistance to disease if a new virus enters the village and it can infect most of the people in the village, and you're different and have a different genotype than most of the people, then you have an advantage. So the one thing that's spreading through the rest of the population, you're immune from. And you can see the effects of this, of the selection for diversity in terms of disease resistance in the HLA region, which is critical for immune response to pathogens. And if you just look on chromosome 6 at the density of SNPs in that region, it's much higher there than elsewhere in the genome, because there's a lot of selection. Obviously, we're constantly exposed to different kinds of infectious disease, so there's a lot of selection for having diversity there. And there's the HLA region, if you couldn't guess where it was. And this is actually an example of frequency-dependent selection. That is, whether a particular allele is advantageous depends on how frequent it is. When it's rare, it's good to be different, so that's advantageous. If it increases in frequency, then it can become less advantageous. And also, it obviously can vary quite a lot with your local environment, what pathogens happen to be nearby or what predators. In humans, it's mostly pathogens we worry about, not so much predators these days. So it can fluctuate quite a bit on small geographic scales and on short temporal time scale. As a side note, pathogens also evolve, and they do some similar things. This is one of the chromosomes of Plasmodium falciparum, which causes the most severe kind of malaria, and where that sharp spike in diversity, it's just showing diversity across the chromosome, that sharp spike in diversity is a gene that codes for a protein that's exposed to the immune system. A lot of malaria proteins are not exposed because they hide in red blood cells, but this protein is exposed to the human immune system. And again, there's a lot of pressure to be diverse, so that if you're the first parasite entering a village, you want to be different than all the other parasites that the population's been exposed to, so that you can happily infect people without having that unpleasant immune system triggered immediately. And they evolve faster than we do, by the way. So they're constantly evolving as well. So that's balancing selection. Purifying selection is the most common kind of selection. It's sort of a little dull because all it is it's the removal of new deleterious mutations. Most organisms are pretty well adapted to their environment, and a functional change, something that changes their phenotype in an important way, is probably bad for them. And it's going to be eliminated by natural selection. It's not going to be passed on for very long. And it's very clear to see this in human genetic data. This is a plot of the top figure here. Plot is from 1,000 genomes data. It's the diversity across the whole genome, looking at different parts of genes. And I've marked where some exons are. This first exon, the middle exon, and the last exon within genes throughout the genome. And you can see the diversity is much lower within these coding exons because changes to a protein are probably bad, and they tend to be eliminated. And you can see that the effect is strong. You can just read off from there how many mutations have occurred in these exons and that have been eliminated by selection over time. And although they're eliminated, they may not be eliminated immediately. Obviously, if something makes you non-viable, then you're not going to see it in the population, but lots of mutations are mildly deleterious. They might make you more likely to be sick or stupider or less attractive, whatever, which might be bad in certain circumstances. And so you can see these alleles are hanging around in the population, but they won't rise to very high frequency because they are bad and they're less successful. So if you just plot the allele frequency of non-synonymous mutations and compare it to synonymous mutations, that's what I show here on the left, as a function of frequency in the 1% bin, that first bin, you see there's an excess of non-synonymous mutations compared to synonymous mutations because more of these are functional, and so that excess represents mildly deleterious alleles that are going to be eliminated eventually by selection, but haven't been eliminated yet. Even within the non-synonymous, you can break it down further. On the right is plotted different categorization of non-synonymous mutations as to whether or not they're likely to damage the protein, change the effect of the protein. So in pink and red, those are changes that have a pretty good chance of changing the protein's function, and those are the ones that are you see more of them at low frequency. So those are the ones that are going to be eliminated. So in terms of evolutionary biology, these are not interesting. This is kind of sludge that's being eliminated by natural selection all the time. In terms of medical genetics or human genetics of disease, these are probably a lot of the ones that we're interested in because these are one of the ways of being deleterious is it makes you more likely to get sick. And so these are some of the things that are causing genetic diseases or increasing your risk for early onset diseases of various kinds. So they're of great medical interest in some cases, but not of tremendous interest to evolutionary biologists. The effect of purifying selection can be seen not only in the allele itself that's being selected against, it can also be seen on some of the surrounding variation. The effect of purifying selection is that it reduces diversity in that region. And you can think of like there's a gene where deleterious mutations keep happening. When a deleterious mutation happens, the chromosome it happens on is going to be removed from the population eventually. And so effectively it's not part of the population now in terms of the long-term success. So you have right around genes or other functional elements, you effectively have a smaller population size. And that means you can sustain less diversity because some of the diversity gets taken, any diversity that's on that chromosome that gets the bad mutation is going to be removed from the population. And it's very easy to see this too. This again is 1,000 genomes data. Here they plotted the distance from a gene, from the start or the stop of a gene across the whole genome. The three colors are three different populations. And you can see, and what's plotted is diversity. And there's a significant dip in diversity around a gene. And it's on the order of 50 or 100 kb. So it's a substantial stretch where there's notable reduction in diversity. And this is affecting the distribution of diversity throughout the genome. So this is an ongoing effect.
Steve, why is there such huge differences? Is it related to the ancientness of the population, that the Europeans have gone through a more purifying selection and the others have not?
Why is the absolute level? Well, there's just less diversity. And the red and the blue are the two non-African populations. And there's just less diversity outside of Africa because they pass through a bottleneck leaving Africa. And so there isn't that much there. There may also be a small difference in how much diversity is reduced there. There's a question of whether purifying selection has been less effective outside of Africa because the effective population size was smaller. But that's a pretty minor effect. And finally, there's positive selection, which is what we usually think of as selection, as natural selection. It's what Darwin was famous for. It's the basis for adaptation, pretty much all adaptation, we think. And so it's kind of the sexy thing to look for. And people have looked for it. I'm going to focus initially on selective sweeps, which is selection where the mutation starts out basically as a new mutation and increases in frequency. I'll add a few complications later. And so the question is, what's the effect of positive selection on genetic variation? And basically that means how can we detect it just by looking at genetic variation. And so let's take the case where selection doesn't happen. This is neutral evolution. So suppose there's a mutation that happens in this blue guy here. And as time goes on, the frequency of that allele, it may increase, it may decrease, it kind of bops around a little bit. It drifts. And the technical term is it drifts. Experiences genetic drift. But on average, it doesn't actually change in frequency. So very slowly it might change over time. If there's positive selection for something, if this new allele, this mutation provides a benefit, well then over time it can rapidly increase in frequency. And that rapid increase is what leaves the genetic signature that we can look for. Actually leaves a number of different genetic signatures. And I'll kind of describe some of them. So let's consider what the genetic situation looks like before selection happens and after. So this is a sort of a cartoon version of some chromosomes in the population. This new red mutation is beneficial. And there's a lot of diversity there. You have different alleles, you have different combinations of alleles, different haplotypes present in the population because it's just been sitting, it's just been behaving normally. After selection has happened, this allele has increased rapidly in frequency and is present in a large fraction of the population. And so the signatures we can look for, well, one signature happens, suppose this selection has happened only in one geographic region, like it happened in, the Boston area. Something that's positively, being a Red Sox fan, maybe it makes you, if there's an allele for that, it might make you more reproductively successful here as compared to New York. If that's the case, then you will find that allele at very high frequency in that region and very low frequency elsewhere. So it'd be unusual. One signature for selection then is that there's an unusually large difference in frequency between populations at that locus. And this can indeed be seen. Here, I just mentioned that a common way of measuring frequency differences between populations is a statistic called FST, but there are other statistics too that you can use. And a classic example of this is the Duffy null allele. It's the Duffy protein is a blood antigen, one of the many blood antigens, sits on the outside of red blood cells and does something or other, not entirely clear what. But one of the things it's not intended for, say, is that it's also the way by which Plasmodium vivax, another cause of malaria, enters red blood cells. And so it's the only way of entering the red blood cell. So it's critical for invasion and for infection. And if you lose that protein, then you are pretty much immune to vivax and malaria. And there is a mutation that knocks out that gene, it's called the Duffy null allele, and plotted, shown there is the density, the frequency of that mutation around the world. And you see it's at very high frequency in sub-Saharan Africa. And as a consequence, people in sub-Saharan Africa are largely immune to vivax and malaria, so much so that there's almost no vivax and malaria across Africa. So this was a highly successful case of natural selection in humans, suggesting that there was a very large cost from vivax and malaria at some point. And outside of Africa, it's basically not there at all. And so this is sort of a classic case. This is, in fact, I believe this is how it was discovered that this allele, that this protein was the invasion route for this parasite. So it can be very useful to be looking at natural selection. A more recent case, in terms of studying it, came from people who were looking for adaptation for handling high altitude, where there's very low oxygen. And so what this research group did was they compared allele frequencies between a Tibetan population, a sample from Tibet, and a Han Chinese sample. Very closely related populations, very similar allele frequencies. So plotted on the two axes are the Tibetan frequency and the Han Chinese frequency. And you can see almost everything has pretty much the same frequency in both populations. And there are two alleles that are at very high frequency in Tibet and not at very high frequency in the Han Chinese. And they're in the two in the lower right-hand corner. There are two alleles in the same gene, EPOS1, and it turns out this does indeed confer adaptation to handling the low oxygen levels present there. So this was a very easy way of finding this particular gene. So in principle, this is a powerful way of detecting where selection has happened regionally. In practice, there aren't very many sort of low-hanging fruit like that. Somebody took the trouble of comparing, how many real outliers are there. They took a whole bunch of different population pairs. So each one of these dots is a pair of populations. And on the x-axis is the average difference in allele frequency measured by FST between those two populations. And on the y-axis shows the most extreme single allele in that pair of populations. And most of the time, you can predict really well what the extreme is going to be just from what the average is. So all you're seeing is sort of the tail of the distribution up at the high end. There are a few cases up here where there are clearly outliers. The one I was just talking about, EPOS1, is one of them. All the other colored dots here are known pigmentation genes. So it's not... There's actually information in there, but by itself, it may not be a very easy way of finding out what's been selected. All right, so that's one. That's just the first signature. I won't talk as much about all of the others. Another signature is low diversity. If everyone or a large fraction of the population is the same now around this new selected allele, well, that means if you're the same, you're not different. There's not a lot of genetic diversity there. And so if you just plot genetic diversity across a chromosome, you'll find regions where diversity dips because there's been a selective sweep there. So what that means is that, I said purifying selection produces reduced diversity around functional elements. Positive selection can also produce reduced diversity. So they have somewhat similar signatures here, which is inconvenient. The loss of diversity can be more profound in the case of positive selection, because if a sweep goes all the way to fixation, everybody's the same, and so there's virtually no diversity present. Another thing that's a little bit easier to look for, and it's sort of a different way of looking at the same thing, is that if everyone's the same, you have the same haplotype there. Basically, if you can predict, if you have that red allele, you can predict what other alleles everyone will have in the population, up to the point where recombination has broken it down. And if this selection happened recently, then recombination hasn't had time to break it down yet. And so you can look for long haplotypes that are high frequency in the population. And there's a whole series of statistical tests that have been developed for detecting that there's an unbroken long haplotype present. And this turns out to be a very powerful way of detecting selection that's happened within maybe the last 20,000 years, roughly. And here, a very, very clear signature for a signal of selection can be seen at one of the now poster children for selection in humans, which is lactase persistence. The normal state for mammals is that as you get older, you lose the ability to digest lactose because lactose is present mostly only in milk, and normal mammals don't drink milk as adults. But in human populations where they've practiced herding for a long time, many adults can in fact digest lactose. They're lactose tolerant. Most Northern Europeans, I'm lactose tolerant because my ancestors are from Northern Europe. And if you look around the lactase gene, there is an enormously long haplotype that's hardly broken at all. And in Northern Europe, it's around 70% frequency. It extends for more than a megabase as a result of very strong natural selection, positive selection, for this trait. And the mutation that gives this capacity for digesting lactose is in fact on that haplotype. This plot just shows that if you plot the strength of the length of the haplotype versus the frequency, lactase sits way up here on the right. It is the strongest signal of selection by this test in Western Europe. Okay, finally, one other signature that's a little bit harder to visualize is that in regions that have been undergoing recent positive selection, there will be an excess of high-frequency derived alleles. Normally, the derived allele, the newer allele, is at low frequency. It tends to stay at low frequency. But when this kind of sweep happens towards higher frequency, it can bring any other mutations that are nearby to higher frequency. And so it can bring more of these rare mutations up to high frequency along with it. So that's a different sort of thing you can look at in data. And all of these signatures have been looked at. They've been known for a while, and they've been looked at in various, looking throughout in scans across the genome, looking for places where selection has happened. And you can do some interesting things with them. It turns out that there's independent information in each of these signatures. And so one thing you can do, and it's an approach that was pioneered by Pardis Sabeti here, is combine the information from the different signals. And here, in a cartoon version, we've got evidence from a long haplotype, evidence from derived allele frequency, and evidence from differentiation between populations. And they're giving you different information, but the actual selected one, where selection actually happened, you can get enhanced information about that, which is important because some of these signatures tend to cover very large regions of a chromosome and not really tell you very much information about which allele or even which gene was actually selected for. So if you want to get down to details, then it's better to combine information. And here's an actual case. I think this is chromosome 5 in humans, and I don't remember what data set this is. It might be 1,000 genomes. These are different signatures of selection. The top one is a long haplotype. Second one is population differentiation, and then the bottom one is derived alleles. And a lot of these are raised or elevated, so somewhere in here there's evidence that selection happened, but they're very noisy, and it's very hard to know exactly where selection happened. But if you combine them, then, as if by magic, the bottom distribution shows the score for where you think selection happened. And I don't know if you can actually see it, there are only a handful that have an elevated score, indicating that right here is where selection probably happened. And it turns out the biggest signal there is a non-synonymous mutation, so there's a good chance it's functional. It's sitting in a gene that's important for skin pigmentation. This, in fact, is an allele that contributes to European skin color.
It's one of the major alleles for that. Yeah.
What kind of sample sizes do you need in order to detect these kinds of selection sweeps?
Good question. Depends on how strong it is. I'd say for really strong signals, 100 is fine. For as you're getting to more subtle things, thousands are better. Above that, the problem isn't so much sample size as knowing, being able to distinguish between background stuff and what's actually selection. It's not a statistical problem. It's basically what we're doing is we're looking for the weirdest part of the genome, and lots of weird stuff can be happening in the genome. And there are various things that can confound these. For instance, the long haplotype test, that can be founded if there's an inversion where that suppresses recombination in that region, and that can that's one of the sorts of things that can confuse you. So there's a variety of things that can affect you. So I said these tests have been known for a while. There was a big spate of genome scans about 10 or 12 years ago as data, genome-wide data became available. But there is still work going on in this area. This is a figure from a paper that was published 2 weeks ago in Science, which a new signature for selection, for recent selection, which I thought was cool enough, but I'll try to explain it to you. The basic idea is that so we've got 2 alleles here at a site, each of along the bottom. Do I have a pointer? There we go. Along the bottom, these are all the samples, and if you just construct a gene tree of which samples are related to which at this particular locus, this is the gene tree. The idea is that this in blue here is a derived allele that a mutation happened at some point in the past, and it's been selected for. And if it's selected for, it's increased in frequency, and what that means is that it's younger, that on average you have more recent common ancestors. And if you compare people, they have a recent common ancestor. That's what it means for it to have increased in frequency recently. And so if you just look at the terminal branches, the terminal lines here leading to all of these samples, they tend to be longer for the one that wasn't selected for because they're older. And any mutation that occurs on one of these is going to appear as a singleton in your sample. That's one mutation. So all this test does is count how many singletons there are around each. You look at an allele in the genome and just count how many singletons there are nearby, and look at the density of singletons. And the density will be lower, fewer singletons around recently selected things. And one of the nice things about this test is that, unlike some of the other tests, I'll say in a minute, it works on not just selective sweeps, but it works in some other more complicated situations. And it's, according to the authors, who they figured out, what is this sensitive to? If you have a reasonable sample size of like a few thousand, then it's probably sensitive to selection that's occurred in like the last 2,000 years. So in genetic terms, that's very recent selection. So this is cool, and it's worth reading that paper. Okay, so as I said, there are some complications. A selective sweep is nice and pure. You have one, but it's making some assumptions. It assumes that the beneficial mutation occurred once, or at least was so rare that you can treat it as there was just the one copy. And then that rises to frequency and has all these effects. And it assumes there's just that one. It's one mutation of pretty large effect. But selection can happen in other ways. Selection can occur on standing variation. That's variants that have been in the population for a long time. If it's been there for 500,000 years or 1 million years, recombination's been happening all the time. So that variant is sitting on lots of different haplotype backgrounds. They're all going to rise in frequency. You're not going to see most of these signals. The same mutation can happen more than once. That's happened in the sickle cell trait. This identical mutation has occurred on multiple occasions, been selected for, but you then have multiple haplotypes increasing in frequency. And finally, there may be lots of different alleles that contribute to the same trait, and each one may only shift a little bit in frequency, and you can still have a substantial effect on that trait. And so these signatures of selection are pretty much hopeless. There are attempts to use sort of modifications of these tests for looking at allele frequency distributions in the case of selection on standing variation, but it's just harder. The signal isn't as easy to find. You can still do some interesting things, though, and find some interesting stuff if you stop thinking just about alleles, but rather think about a trait. If you know what genes or what alleles are contributing to variation in a trait, then you can aggregate different alleles that are involved in that trait. And so this is an example that was carried out by Joel Hirschhorn's group here in Harvard, and looking at height, stature in Europeans. We know from a GWAS many of the variants that affect stature. We also know just from looking at Europeans that there's a climb in height across Europe from north to south. Northern Europeans tend to be taller than Southern Europeans. And so what they did in this figure, they ranked the SNPs from the GWAS in terms of how large an effect each SNP had on stature. And then on the y-axis, they plot the difference in frequency between Northern Europe and Southern Europe. And what they find is that the alleles that have the biggest effect on stature are also the ones that have the biggest frequency difference in Europe. And this is consistent across all of the major alleles here. They all have higher frequency in Europe. This isn't just sort of randomly a few alleles happen to be higher. All of them have higher frequency in the increased height direction in Northern Europe. Or alternatively, they all have the lower stature allele in Southern Europe. And this is pretty good evidence, and it's been substantiated by further studies by others, that selection was acting on stature in Europeans in some way. Not clear exactly how, but in some way. And there have been other studies. This is a similar sort of study looking at a variety of traits correlated with the geography. In this case, finding a significant selection for stronger response to damage from ultraviolet radiation if you live near the equator, if your population comes from near the equator, which maybe isn't too surprising. But the different colors are different continents, and on different continents, the same sort of selection pressure has been happening. So you can indeed extract a fair bit of information about the trait that's been selected for, even if you may not necessarily know which particular allele has been selected. So that leads to the question, what traits have been selected for? What have we actually found? What's been going on in humans in the last 20,000 years or so? Because that's pretty much all the data I'm talking about are from selection that's occurred within the last 20 to 40,000 years. Because that's the easy place to look. Lots of interesting selection happened before, like what made us anatomically modern humans, but that's a lot harder to find, a lot harder to study, so we're studying all the relatively easy things. So as I said, you can scan the genome for these signatures of selection, and many people did. Here's the results of one of those scans, because it was one done here. And here are places in the genome where there's evidence that selection happened. There are probably some false positives in there, but there are lots of actual cases of selection that occurred there. And the question is, what do they do? What was involved? And the answer is mostly we have no idea. Something happened there somewhere, and we don't know what the trait was. We don't know what the selection pressure was. And it's just sort of going from something happened at this locus to what's the phenotype is hard work. That's biology. That's not sitting around looking at 1,000 genomes data. You actually have to do some biology then. And I'll have one example here of the kind of work that can elucidate this sort of thing. This is a little segment of one of those genome scans. This is chromosome 2 in East Asians. And this is a test for long haplotype. So it's very high score here across this entire region, indicating that selection happened here. Where exactly, probably somewhere in here, but you can't really tell. Yeah.
Long relative to whom or to what?
Long relative to, say, the size of a single gene. So a long, and also the sheer number of variants here is a problem. If you want to see, was one of these variants had a phenotypic effect, actually drove this, because one of these variants was probably actually important, and the rest didn't. So figuring out, knowing what to test for, say, if you want to do some functional work, this by itself is kind of daunting. This is just too many variants there to put into a model organism, say, and see it more into a cell line and see what it did. But you can do the trick I mentioned before of combining information from multiple signatures, and that actually cleans up this particular locus very nicely. There are only a handful of then candidates you want to look at, and one of those turns out to be a non-synonymous change. As I say, you can see that, here you can see that there are in fact multiple genes across this region. And it was a non-synonymous change in a particular gene known to be involved in development of hair and sweat glands. It's known to have been under selection in other organisms. So that then gives you something you can look at in more detail. You can take that variant and study it in the lab. And somebody did, a postdoc, Harvard, working with the Broad. And as I said, this was selection that apparently happened in East Asia. This increased in frequency, and she stuck it in a mouse. This particular variant stuck it in a mouse and saw what it did. And, well, it did several different things. It produced thicker hair, it produced smaller and denser mammary glands, and it produced a higher density of eccrine sweat glands. And it turns out that at least the first and third of these, the hair and the sweat glands, this has the same phenotypic effect in humans. And the thicker hair is actually, is something you can actually see. East Asian hair doesn't look the same as European hair, say, typically. So this is a mutation that was selected for. It's illustrative in that by focusing in on something that was selected for, we found something that clearly had a notable phenotypic effect on humans that distinguishes between humans. It also illustrates one of the problems, which is that mutations don't always have one effect. And so it affected hair and affected sweat glands, and we don't know why. You can guess, well, maybe it had something to do with temperature regulation, sweat glands were important, or maybe hair was important. We don't know. So it's not, a final answer, but it was a major clue to finding something that's phenotypically important and that distinguishes humans from one another. If you look more broadly, this is from a very recent review article by Sarah Tishkoff's group in Science of what we've learned about regional selection in humans. And you can see on the map we've learned a number of different things, and several of the cases I've talked about are on this map, and I'll just mention them, like selection for changes to diet, the lactase persistence case up there in Europe. That's one of the classic ones. If you can't find lactase, then you're doing something wrong and you're studying selection. But that's also been studied in East Africa and the Middle East, where there have been other herding populations. Turns out to be independent mutations that in the same regulatory region that have the same phenotypic effect. So it's exactly the same pathway, exactly the same mechanism, but that's occurred multiple times in different places. There are other populations around the world that have lactase persistence in South Asia, in West Africa, and these have not been studied in any depth at this point. So exactly what the mechanisms are there is as yet unknown. I mentioned skin pigmentation. There are a bunch of genes that are known where alleles are known to contribute to the paler skin color that Europeans have, and which varies with latitude. And so they've been well studied. Other genes are known to have mutations in Asia. So some of them independent, some of them are shared. So it's but a partially different set of mutations. Other parts of the world where there's also variation in pigmentation, like within Africa, there's quite a lot of variation, and probably some of that has been under selection. But again, the studies haven't been done there, because there's plenty of things still to study. I mentioned the polygenic selection on stature in Europe. Turns out there's also been selection on stature elsewhere in rainforest environments. There's selection for smaller stature in humans. Pygmies that tend to have small stature, well known in the Central African rainforest. And selection there is operated in a rather different way. In Europe, it was very polygenic selection on lots of different alleles. In Central Africa, it apparently was strong selection operating on a handful of loci for shorter stature. Might be because the pressure was of a different kind. It may just be that's the way it happened to work. Don't really know. And I think the last one I circle here is the one for high altitude adaptation. That's been studied in Himalayas. It's also been studied in other high places, in the Ethiopian highlands and in the Andes, and you will find, again, independent mutations, most of them in the same pathway, leading to similar phenotype. So same selection pressure, different mutations, but similar outcome.
What's the thinking about the selection for short stature? What's the advantageous nature of that?
I don't know much about the thinking. It might just be a matter of resources, of you're better off, if you need to eat less, because it's not a very highly nourishing environment, but I don't really, it's not something I've looked at in any detail. So those are the cases we've understood something about. We don't always necessarily know exactly what the selection pressure was, but at least we have some idea of what the phenotype might be. One question that might occur to people is, well, how much of this is there? How much has natural selection been operating in humans recently? And it turns out to be kind of tricky to figure out because, all right, so there's differences, frequency differences between populations. It varies across the genome. How much of that is naturally occurring, and how much of that is neutrally occurring? How much of that is the result of selection? Especially since we don't really know the demographic history of humans in detail, we can't exactly model it and tell you what the distribution should look like. So it's kind of an open question of how much positive selection has happened. And one way of trying to address this is to look at that reduction in diversity, like particularly reduction in diversity around genes. And as I said, that can be caused either by selective sweeps happening repeatedly, positive selection, or by background selection. So you need to find a way to distinguish them. And so one group, what they did was they looked at genes, so they're only looking at genes, so there's presumably background selection that's happening all the time. And they compared cases where there's been a non-synonymous change in humans over the last million years or so, to places where there's only been a non-synonymous change. And the idea is if there have been a lot of selective sweeps, you should see reduced diversity around the non-synonymous case. And here's their plot from their paper. This is Hernandez et al. From 2011, so it's a few years ago now. Here's where the substitution happened at some point. And they plot diversity for synonymous in blue substitutions and for around non-synonymous substitutions in red. And those distributions are basically the same. There's no obvious difference between the non-synonymous and the synonymous. And their conclusion was, we don't see any evidence for lower diversity around functional changes. Classic selective sweeps have not, there have been some, but they're not they're not a major factor. Maybe it's been, this polygenic selection, other kinds of selection are happening. And basically said in the conclusions, you should stop wasting your time looking for these things, which was a little annoying for those of us who were looking for them, in particular those of us who wanted to get funding to look for them, because then, the reviewers of the grant proposal say, why are you looking for this? Hernandez et al. Just showed that there aren't any selective sweeps, or you've already found them all. Don't waste your time. And then a couple years later, there was another paper from a different group, David Enord, Dmitry Petrov's groups, and they came to a very different conclusion. I think I have time to go through this. And they said, they looked at the same data and they said, no, you've drawn the wrong conclusion. Because the problem is, you're looking at non-synonymous mutations. Well, let's think about that a little bit. Let's think about, you've got two genes. Gene A is highly constrained. Every amino acid is a precious jewel, and if you change one of these, it's bad. So anytime there's a missense mutation that's deleterious, you will find very few missense mutations that happen neutrally. So if there's any missense mutations, they will have been beneficial. And what that means is lots of background selection happens. There's lots of purifying selection going on all the time because all of these mutations are bad. So if you look at diversity around that gene, well, there's going to be very low diversity around that gene. Now, gene B, on the other hand, this is a weakly constrained gene. There's a lot of the amino acids don't do anything. So there'll be a lot of neutral changes. And in consequence, there's going to be less background selection because there's fewer opportunities for deleterious mutations. And so around those genes, there's going to be a smaller reduction in diversity. And what they say is that when you look at non-synonymous, you pick a non-synonymous substitution that's happened, which is what the Hernandez et al. Group did, you're picking primarily from this group, from these genes. On the other hand, when you're picking non-synonymous mutations, you're picking from both of them. So you've biased yourself to finding places where there's not a lot of change. And so if there are a lot of cases of positive selection going on, selective sweeps, you're going to completely lose it because of this bias. And so their conclusion was that there's actually strong evidence for lots of selective sweeps in recent human history. There's been a high rate of strongly adaptive substitutions near amino acid change, and there's been even more sweeps that have been driven around regulatory changes, which I think we know independently. So the sort of same data, very different conclusions, and a little heartening to those of us who are interested in selection. I mentioned that I introduced that new test for selection, is this density of singletons. They conclude that lots of different traits, if you just accept their data at face value, lots of different traits, quantitative traits in humans that have been studied by GWAS, a lot of them show evidence for having been undergoing selection in the last 2,000 years, like more than half. I don't know if I believe that, but it's certainly there's evidence that suggested that lots of selection of some kind is going on all the time. Okay, so that's where we are in terms of studying selection from just looking at genetic variation and what its effect is. But there's one other topic that's really interesting because it gives us a whole new way of looking at this, and that's ancient DNA. Because in all these things, we're inferring what happened in the past in these kinds of studies. We look at genetic variation in a bunch of Norwegians, and you try to figure out what happened 10,000 years ago. But now we can actually look at DNA from 10,000 years ago or 5,000 years ago and see directly what's changed between then and now. And people have done this. We now are at the point where we've sequenced enough ancient genomes, we can compare allele frequencies in the past to allele frequencies now. And this is data from a paper from last year, David Reich's group at Harvard. And this is plots, the colored dots are the frequency of several particular alleles that are of interest in several ancient populations. And the dashed lines are what the frequency is in modern populations, modern European populations. This is basically Europe and West Asia are these ancient populations. And I'll point out a couple of cases. The top left one is lactase. Again, this classic example. You can see that the frequencies in these ancient populations was at or near zero, the allele that gives you the ability to digest lactose. Whereas in modern Southern European populations, it's still fairly low, but in Northern European populations, it's very high. So this is selection that's happened just in the last few thousand years. You can see that it's been dramatically increased in frequency over that time. Here, on the other hand, is a pigmentation allele, one of the major ones that contributes to European paleness. And you can see that was at different levels in different populations. They were apparently the steppe peoples. These are the people of the western steppes in Asia. They were apparently probably paler than these other Europeans down here, but they're all a lower frequency than in modern Europeans. So this is a selection that was ongoing at this time and has continued into the present. And if you look across the genome, this is I don't know if you're used to Manhattan plots, but these are the entire genome here spread out, and the places where there are signals that selection has happened just from comparing the ancient DNA to the modern DNA, you find a lot of the same things. You find that there are skin pigmentation genes that are already known. You find the diet genes, including lactase and like fatty acid dehydrogenase, which has been seen to be under selection in other populations. Selection for resistance to infectious disease at the HLA, at toll-like receptors. So the basic story we get from the ancient DNA is it's very similar, which is heartening because we were reconstructing the past based on computer models, and it's nice to see that when you actually can look at the ancient DNA, it tells you we were right in a lot of these cases. We were really correctly inferring that selection happened. But you can get a lot more detail when you look at the ancient DNA, because our computer models are simple. So I'll give you a few examples of the difference in the story and to end here. I talked about this gradient of stature in Europe, and there's this north-south gradient, so we concluded selection happened. Well, when looking at the ancient DNA, they concluded a little more detail. They concluded that selection happened for shorter stature in southern Europe, that selection for taller stature happened in West Asia in these steppe populations, or at least that's where we see it, and that we see this greater height in Northern Europe because those people then moved into Northern Europe. It wasn't necessarily selection happening in Northern Europe for a greater height, it was just people happened to migrate in. And looking at modern population, we would have no idea about this. It's very hard to figure out all of these, where all these people have been moving around. We tend to assume that, well, if we're looking at Chinese people today, that's who, their ancestors were living there 10,000 years ago. No, people move. They move a lot, it turns out, when you look at ancient DNA, sometimes in ways that we couldn't tell at all from modern DNA. All right, the second case, the pigmentation. There are two main genes that contribute to European pigmentation, typical color, with very similar names, SLC24A5 and SLC45A2. And we know from the genetic evidence, modern Europeans, that they've both been under strong selection. There's an allele that's risen to high frequency in both cases. Turns out the two genes have somewhat different histories looking at the ancient DNA. One of them, actually it's the one I just showed, this skin pigmentation gene, you can see rising in frequency within Europe as selection was occurring for lighter skin. The other allele actually entered Europe at very high frequency with farmers. When the first farmers moved in from Anatolia, it's now Turkey, they largely replaced the European population, and they already had lighter skin, presumably as a result of earlier selection. But you get a much more detailed picture of the history this way. And finally, the last case is the case of EDAR, the one thing that gives you more sweat glands and thicker hair. Well, when we were studying it, the story seemed pretty simple. This plot shows where that allele, this selected allele is. It's present in East Asia, and it's present in the New World because the people from Eastern Asia populated the New World. And so it was clear, you can do they did very detailed modeling for this, computer modeling, and it was clear, this is estimating where this allele originated. And it originated in central China 30,000 years ago. So it was a nice, simple story. It was a Cell paper, and it's a great paper. But this particular conclusion, the problem is you look at the ancient DNA, it turns out this allele was at high frequency in Swedish hunter-gatherers 6,000 years ago, which is not something you would guess from looking at the modern distribution. And it says, it was under selection in East Asia, and that story hasn't changed. But the details of what happened historically, something more complicated happened. It probably arose in Western Asia and happened to be selected for in Eastern Asia. And this is the kind of information we get from really a tiny snapshot of ancient DNA. As we get more DNA, we'll be able to learn a great deal more, at least from places where there is ancient DNA. A lot of the world, in tropics, DNA does not preserve well. I'm going to conclude with a couple of comments. So recent positive selection, it clearly has had an impact, a significant impact on human phenotypic diversity, both individuals and within groups. Exactly how much, some traits, many traits, at least two traits, have been changed by this, and many of these traits are of medical interest or biological interest. It's a great way of finding out, one way of finding out where these important phenotypic changes are. So if you study natural selection, you can identify places where things have changed. It's not by itself, it's certainly not an all-purpose tool. It's a clue. It has to be combined with functional work, with GWAS, with association studies, with all kinds of other things. But it is one tool in the toolkit. And I'll stop there. Thanks.